So the reason why we chose this as the first topic that we would cover this morning is because, as I'm sure you've seen, the really very exciting news in the media that has been over the last few months that the European Medicines Agency recommended licensure of a group B meningococcal vaccine, the first one to be licensed for use from early infancy throughout Europe, just before Christmas, and that was then ratified a matter of weeks ago by the European Commission. So we now, for the first time, have a licensed vaccine for this disease. If you go back and look at the vaccine programs, the program that was in the 1990s, and I can see there's quite a few people in the audience who remember even the 1980s and 1970s. Looked like then, which was rather simple. It was quite easy to remember this. By this year, the program's got a bit more complex here. And as we go through the morning, you'll hear about some additional antigens which are going to be added to the vaccine schedule for children this year. And I'll show you, I think at the end of my talk, what we hopefully by Christmas time you'll see as the schedule here. But the reason for focusing on this disease is it doesn't cause a huge burden of hospitalisations that influenza and bronchiolitis cause, but it is the leading infectious cause of death in young children. And for that reason has been an absolute priority for public health development, in fact over the last 20 years. If you look under the electron microscope, have we got a pointer? Anyone know? So if you look under the electron microscope, that's what this part of the meningococcus looks like. And those spots on it are, in fact, tiny particles of gold that we've stuck onto antibodies that are picking up proteins on the surface of the bug that we can detect. And I'll come back to that in a moment. So the bug is absolutely decorated with many different types of proteins on its surface. But surrounding that is a sugar coat, a capsule made of polysaccharide. And there are different polysaccharides on the surface of meningococci which allow us to define them into different names. So most of the disease in the UK is caused by B, and that just means that they're meningococci with sugars that are called B, which is a particular collection of sugars on the surface. And in the 1990s it was C, which is why we introduced the MenC vaccine in 1999. And you can see here in the dark blue the amazing impact that's had on disease caused by MenC. So in those who are vaccinated, if you remember, it was everyone up to the age of 19 years, disease just completely disappeared. And so there you can see in dark blue. And in fact, we think there's somewhere around 15,000 cases have been prevented of this awful disease just since 1999. So it's an incredibly short period of time. And of course, around 10% of those cases would have resulted in death. So it's been an amazing success, the introduction of that vaccine. Now, one of the reasons why it was so successful is that these bacteria live in our throats, and the vaccine that was introduced in 1999 blocked transmission from person to person of the bug. And that means that those of us who, thank you very much, that those of us who were carrying the organism but weren't suffering from any disease also could no longer carry it. So even the people who weren't vaccinated were no longer getting exposed. And you see that on this graph here, that these are older pensioners, in other words, everyone over the age of 19 years, and you can see that disease has also disappeared in that group. And that's because by immunising children, we stopped anyone from carrying it, stopped all the children from carrying it, and therefore they could no longer transmit the bug to other people. And that meant that since they were the main carriers, the under-19s, that all the rest of us were protected just by immunising children. And you can see that here, the vast majority of carriage, which are these circles, happens in adolescents and young adults. And those are the ones who have very high levels of immunity. And I'm going to just make a very brief comment here, and that is one of the changes to the vaccine schedule this year will be the introduction of a teenage booster of MenC vaccine. And the reason for that is that we currently vaccinate very young children, but after they've been vaccinated, the level of protection falls quite rapidly. over the next few years. So that we've got this group of children now growing up who, although they were protected when they were very little, are no longer protected against MenC. And we know that this is the period where disease happens. It was in very young children who are protected at the moment from vaccine and teenagers. So by putting in a booster dose of vaccine here, we'll be able to keep the levels of protection, levels of antibody high in adolescents and protect them from this period of risk of disease, but also protect everyone else in the population, because again we'll have an impact on carriage in this age group. And here, a couple of years ago, the Daily Telegraph advising the Department of Health on policy that teenagers should get a booster dose. So it's expected that this will happen from September, and we'll also reduce the infant schedule to 1 dose. So then our MenC schedule will be 3 months, 12 months as part of the MenC vaccine, and then a dose for adolescents. Now there are, for some of those other sugar types of meningococcus, there are already vaccines out there. There's these 3 different ones from different manufacturers, which cover 4 of the 5 main types of meningococcus. But actually we've had very little of AW&Y in the UK. And so at the moment, policy hasn't changed to switch to introducing these to try and give broader protection. We're sticking just with MenC at the moment, but obviously that's being watched very closely by the Department of Health to consider whether changes in the epidemiology of meningococcal disease means that we should at some point in the future consider these vaccines. And in fact, if I show that on this graph, you can see here in red was the disease caused by MenC in the 90s. Vaccines were introduced at the end of the 90s, and you can see the red has almost completely disappeared now as a result of that vaccine. And up here in the green and blue is disease caused by these other 2 types, which is a tiny amount of disease, and that's because it's so little. That's the reason for not changing the vaccine to cover those. But actually the blue here at the bottom is the big problem. This is MenB, which is really what I wanted to talk about this morning. Now MenB, unfortunately, is a bit more complicated in vaccine design. The B does refer to a polysaccharide capsule on the outside of the organism, so a sugar capsule, just exactly the same as with those other meningococci. But the difference with the B capsule is the bug has been very clever in cloaking itself with sugars which are actually sugars that are on our own cells. So this bug, to our immune system, looks as if it's just part of self, looks like it's human almost, because it has sugars that are normally found, in fact, in the brains of fetuses. And so our immune system doesn't make any response to this sugar capsule, and so we can't make a vaccine from it. So the B has been very clever by stealth. It's coated itself with antigens, which are these sugars from humans, so that we can't make a straightforward vaccine as we have done for MenC. And the reason why we're here talking about the excitement of a MenB vaccine that's been developed, which is actually nearly 40 years since people tried to make one, is because it wasn't possible to make one from this polysaccharide. That's why it's taken so long. So because you can't use the polysaccharide, we then have to look underneath the polysaccharide to these proteins that decorate the surface. And then remember I showed you that slide with the little black dots on it, and that's just one of the proteins. In fact, it was a protein called OPA that we were highlighting at that point. And the proteins underneath the capsule, and there are many hundreds of them, are all potential candidates to make into a vaccine, so that our immune system would recognise the protein, and then if the bug then came to our throat, it would be killed because our immune system would already recognise the protein. This, unfortunately, is not as straightforward as it sounds, because these proteins are incredibly variable. And in fact, if you take one of our favourite proteins, which is this one, it's called 4A, it doesn't really matter what it's called, and you look at disease occurring in the UK here in the year from 2007 to 2008, this is the number of different variants just of that one protein. You can see that I can't even count them, there's so many different variants cause disease. This is a frequency distribution, so you can see that some cause a lot of disease, but then loads of them just cause occasional cases. And so there's a real additional problem once you get below the capsule, which is on all MenB organisms. You come underneath the capsule, and it turns out it's a highly variable organism. So you couldn't choose just one of these and make a vaccine, because that would only protect against that one case. It wouldn't protect against all these others. And that's been the real problem for vaccine development. Even when you've identified good proteins to make into a vaccine, how to get them to present to the immune system. And the way this has been dealt with over most of the last 40 years has been to try and take this, the surface of the organ, the membrane, and use that to actually fold the proteins. And the way that works is, as the bacteria grows, this is another electron micrograph of meningococcus, it makes these blebs of its surface membrane that it's making all the time whilst it's growing. And you can actually see them under the microscope, here's them very close up. So little bits of the outside being sloughed off to make little vesicles. And you can use various different methods. The most commonly used one is a detergent method for actually forcing these blebs to form nice spheres of outer membrane. As they were so blue, here's some I made earlier. So these are blebs, we call them outer membrane vesicles of the organism. And they contain a full complement of all of those hundreds of proteins that are there on the surface of the bacteria. And over, since the 1970s, people have been trying to make vaccines from these blebs and immunising children and adults with them to make an immune response to the bleb. And in fact, you can show that when you do that, you make immune responses to many of the different proteins that are there in the outer membrane. And in fact, studies have been done in all these different countries, sometimes implementation of vaccines during epidemics. And those blebs seem to have protected between 50 and 80% of the people who are vaccinated. So, and in fact, we've known this now for 20 years that these blebs worked. So the question is, why haven't we just moved forward and made a vaccine of these? So here, they're the most recent large-scale use was in New Zealand. There's a huge outbreak in New Zealand of meningococcal disease lasting, in fact, over nearly a 20-year period. And the vaccine was introduced, and although it looks as if the epidemic was already going away, which it probably was, some very careful case control studies show that the vaccine had an effectiveness of between 70 and 80%, which is pretty good for this disease which has such a high mortality. So you might say, well, we've got a vaccine that works, why didn't we just implement that here? And the reason is this, that these blebs do make fantastic immune responses against the strain that's causing the epidemic. And here's the New Zealand strain. It was a single strain over that long period in New Zealand, causing almost all of the cases. So by making a vaccine from that strain, it was quite easy to break the epidemic and stop the disease. The problem for the UK, as I showed you with all those different proteins, the core A proteins, we don't have an epidemic from one strain. We have disease caused by many, many hundreds of different strains. And here, if we look at whether the antibodies that Pollard made in New Zealand could kill the Norwegian strain, which in the 80s caused an outbreak in Norway, what you find is it doesn't work. And so this is the problem. These blebs are great if you have an epidemic, but for most of the disease that we see around the world, which is caused by many different strains, it's not of any use because it will only work against the epidemic strain and not against the other strains which are circulating. So that's led to the development of two different approaches. One approach by Pfizer, which I'm not going to talk about this morning, that's in exciting advanced stage of development, and that hopefully will be something we'll see more of in the future. But I thought I'd focus for the remainder of the talk on the vaccine which has just been licensed in Europe. Now, this vaccine does in fact contain this bleb of outer membrane from the New Zealand strain, because actually quite a lot of our cases over the last 50 years or so have been caused by bacteria which are related to the New Zealand strain. So this is the bleb from the outer membrane, and we think that this particular protein, the core A, which I talked about before, is very important in making good immune responses. So we have that as one component in the Novartis vaccine. But that's not enough, because as I showed you, there are lots of different types. So having that in there helps. And what Novartis did is this was published in 2000. They were involved in a major project, one of the first projects to sequence the whole genome of an organism. And now thinking about this from where genomics has moved, now we can sequence the genomes of many organisms, in fact many people, in a week. Whereas this took many years to just sequence the genome of one bacterium. But because all of the genetic material was known in 2000 from this organism, it was then possible to look at the genome and identify genes which would make proteins that might be important in protection against this. And so they took a completely new approach by using a computer to predict how to make a vaccine. So they went to the genes from the bug and used the computer to predict which proteins might make good vaccine candidates. They identified many hundreds of them which could. They tested a lot of them in mice, and then eventually came up with a small number that looked like very good candidates. And in fact, we've already talked about 4A in the blood, but then they chose these 3 proteins as proteins which appeared to be very likely to provide reasonable protection between the different strains of meningococcal. And so what's now happened is that this has been put together in a vaccine. It has these 4 components, the bleb and 3 proteins. 2 of the proteins actually need to be fused with some other bits of protein just to stabilise them because of this problem of folding that I mentioned when it's put into the liquid in the vaccine. And so here they are, those 3 different proteins. Now, I need to just very briefly digress to tell you how we measure immunity to meningococcal. And it's not like if you were looking at antibodies to tetanus, where it's just the level of antibody that's measured. In the development of meningococcal vaccines, people want not just to know that there's antibody, but also that those antibodies can kill the bacteria. And this is really work which goes back to before the 1920s, when it was recognised that in animals, if you take blood from them, the blood from some types of animals will kill meningococcal, the blood from other types of animals doesn't. And so investigations went on eventually, came to these studies in humans, which were led by George Heist. And in 1922, he found that most people did have activity in their blood, most adults had activity in their blood which killed meningococcal. And he found that if you took, I went around this room and took the bacteria, the meningococcal that are in your throat, in about 10% of you, that those bacteria would be killed by the blood of most people in this room. But there's a lot of variation between people in how well our blood kills meningococcal. Adults are much better than children, for example, even between adults. And in all the experiments that they did, they had one person who was the control, whose blood never killed meningococcal. And here you can see what they did. They had a glass tube, they squirted the blood in, it formed a clot, and they added the bacteria, and the people whose blood killed bacteria ended up with no bacteria alive, and those, when the next day when they opened up the glass vials, and for the other people who didn't kill, then they would be able to culture the bacteria the next day. These experiments were incredibly important because they really set the scene for where we are now in understanding immunity to meningococcal. But the very sad part of this story is the person whose blood didn't kill meningococcal was George Heist, and he died from meningococcal disease just before this paper was published. Really, we have to fast forward now to the 1960s, where the really important information was recognised that if you look in the population, so this is the rates of disease, this is the rates of disease from meningococcal disease in America, and you can see that there's a peak of disease in the first couple of years of life, and then the rest of us don't get much disease, which sort of fits with that idea that most adults have some protection already, and this group of young children don't. What they did is they used this bacterial killing assay to show that at this time when there's all this disease in young children, they don't have any antibody. So this is the antibody levels, and this is the disease levels. Whereas if you come to adults who have no disease to speak of, we've all got very high antibody levels. And using that information, the idea is to make a vaccine that makes these young children look more like adults, so that young children have a higher level of antibody and therefore disease will go away. So to go back to the vaccine, which has these 4 different components, to be able to test them, we need to show that those different proteins are all contributing to the killing of bacteria. And that's turned out to be quite a difficult process, because you have to find strains of bacteria which only contain one of the proteins and not the others, so that you can show that that particular component of the vaccine is important in killing the bacteria. And this has been a major undertaking to show this. And I'm just going to show you 2 slides from very early studies that were done. And in fact here in the Thames Valley, the first ever study in babies of this vaccine was undertaken. And here you can see it was just published in 2010. And this summarises it. So what I'm showing you here are the different bacteria which we wanted to see if we could kill. And these different coloured bars here, I think we'll just for now focus on the red one at the end. These are the antibody levels in babies who have been immunised with 4 doses. And in red is the antibody level after the 4th dose, the booster dose at a year of age. And what I want you to notice is that this is a bacterium which contains one of the proteins. And the antibodies induced by the vaccine were able to kill, in 100% of children, were able to kill the bacteria that contain that particular protein that's in the vaccine. So what you see as you go along, and you just look at the red ones, that if the bacteria that we're trying to kill contains the vaccine protein, then the vaccine seems to be highly effective. And down here is one where there's almost no killing, and that's a meningococcus that contains none of the proteins that are in the vaccine, so you actually wouldn't expect it to be killed. And that gives us some idea that if you had meningococci circulating in the UK that contained the proteins that are in the vaccine, this vaccine would work very well. And so we have the first evidence that against these many different strains that have been looked at, there is some activity, even in babies, of this vaccine. We also looked in other schedules. This is looking at if you went with slightly older infants immunised at 6, 8 and 12 months. And what you see here is even better responses than we saw with younger babies. But again, if you have a strain which doesn't contain any of the proteins, we can't kill it. And in fact, following these very early studies, we went on to lead from Oxford a multi-centre trial across Europe to look in a much larger group of children and how well this vaccine would work. And you can see here that, well, you can't read that, but the bottom line is that in this very large study, the vaccine was shown to be to confirm, we were able to confirm the results in the small-scale studies which were done here at Oxford initially. So this involved thousands of babies and demonstrates that the vaccine does make good immune responses. Now, one of the other questions is how safe is the vaccine? And one of the most important aspects of safety that are documented in these types of trials, it's obviously very rare events you can't determine from clinical trials. You can't do that until you've got much larger studies. But one of the first aspects is, what about local reactions? If we do use this vaccine, what do we expect to happen to these children? And what we can say is that there does seem to be a little bit more tenderness, erythema, swelling, and hardness in the injection site compared with routine vaccines. So you can see here that on the right side of each of these is routine vaccines. This is in fact MenC with routine vaccines, and here is the MenB. So it does cause a little bit more local pain and soreness than routine vaccines do. When you look at more generalised reactions, there's the same findings. It makes babies a little bit more irritable. They don't sleep quite as well as with routine vaccines. And you can see that documented here. Rashes about the same between the two. And perhaps the most important one is fever. Now this is done slightly differently in the way this graph is presented. the panel at the top is the MenB vaccine, and the panel at the bottom is routine vaccines. And what you're seeing is how much fever there is afterwards. And what you see is that after the vaccine's been given, you have some fever on the first day, a little bit on day 2, and generally nothing after that. And that's true whether it's for the first dose, the second dose, or the third dose. Now, routine vaccines do that as well. So the vaccines you're giving routinely in primary care do cause some fever in about 20% of babies on the first day, a little bit left on the second day and on the third day. Again, true for each of the first 3 doses. And what you find is if you give MenB at the same time as routine vaccines, you more or less double that. So the MenB causes some fever and the routine vaccines cause fever. If you give both vaccines together, you just add those on top of each other. So we will expect that there will be, if this vaccine is used, that there will be a bit more fever in babies as a result of that. And that's going to need very careful communication to parents that might happen, and also advice on how to manage it. So one of the really important questions, to come back to what I've shown you, that this vaccine has some good safety data about it. We know that it will make immune responses against strains which contain the proteins. The really critical question is how many of our strains actually do contain these proteins and will be killed by the antibodies induced by the vaccine. And this is very difficult to predict. And the reason why it's difficult to predict is that you can look at all the strains that exist in the UK, and you've got to know for every single one of them whether or not that proteins can be made by the bug. You can do that just by looking at its genes, which today is very easy to do, and we can look at all of the bacteria that cause disease in the UK very easily. And in fact, the Meningitis Research Foundation, who are here today, have funded an amazing project actually providing the genetic sequence of almost all of the meningococci causing disease in the UK in the last year. So that as a unique resource for the world, we now know how variable those bacteria are. But unfortunately that's not enough, because we also need to know whether it's on the surface. And the problem then is, that's whether it's expressed or not, the problem then is it turns out that there are lots of little variants of the proteins. They're not all exactly the same. And some of those variants will mean that the antibodies that we make from the vaccine won't bind. So there's a real headache in trying to predict how well the vaccine will work. Novartis have developed this assay, which I'm not going to explain to you all today, and whether this is correct, we won't know actually until the vaccine is used. But using this assay, they predict that somewhere around 73% of strains will be covered. Now, the problem with this is it's trying to relate a fancy assay to this killing assay, and then relate that to the likelihood of protection. And at this moment, I don't think we have absolute confidence that the results in a laboratory will definitely translate into protection. Obviously, we hope that this figure is true, but we won't know that until the vaccine is implemented and we can look at how much disease is actually prevented. Now, one thing I am confident about is it will prevent some disease. And obviously, as for the leading infectious cause of death, that's a very important impact. Now, it's unknown whether with this vaccine there will be an effect on carriage and whether we'll see herd immunity. Now that the vaccine is licensed, where do we go from here? So this year there will be discussions about the vaccine at the Department of Health and the Joint Committee on Vaccination and Immunisation. One of the important questions is it cost effective? And it's very hard for vaccines against rare diseases to be cost effective, because there aren't very many cases, so you have to spend a lot of money buying the vaccine, but you don't prevent very many cases, even though they're important. And so that will be an important component of those discussions. And it's not known whether there will be any recommendation this year from the Department of Health about use of a MenB vaccine or not. And then once there is a decision, the next steps after that will be planning how it's actually implemented, how communication about fever rates, for example, will be undertaken. So if the data we have from the lab are correct, we might be able to prevent 70% of cases of MenB which is would be a real protocol coup for the UK. We don't yet know who would be the right groups to vaccinate. I think it probably will have to be infants as the population who are vaccinated. And we don't yet know whether the vaccine is cost effective. Those analyses are being undertaken by the Department of Health at the moment. One, I think, really important thing to say, though, is that we've reached a point where we now in Europe have a licensed MenB vaccine. If we can't make a case for introduction of the vaccine based on the evidence that we have, then and the governments around Europe say it's too expensive or we're not confident enough in it, whether it will work, then that will be, I think, for decades to come, the end of development of MenB vaccines, because there is enough data behind this already to say that it could have a big impact. If that isn't enough, then it's hard to think that any other company would be able to provide more weighty data to drive the introduction of these vaccines. And of course, pharmaceutical companies are businesses, so if this vaccine doesn't get introduced anywhere in the world, they're not going to invest in new MenB vaccines. So we're actually at a really important time in vaccine development against meningitis. That if we don't have one of these vaccines, not necessarily in the UK, but driven into usage somewhere in the world, then from a business perspective, I think research and development in this area will stall for decades to come. So I promised you some final pictures on the vaccine schedule. This is the program in the 90s. This is the program in 2011. And this is what we're expecting things might look like at some point in the future with a MenB vaccine introduced. And what's added on here is the dosing of MenB, the addition of hepatitis B vaccine, the addition of rotavirus vaccine for diarrhea in young children, all hopefully to be added during the course of the next, certainly most of these this year, then the next few years. And what I don't think I've got on there is the teenage dose of MenC, which should be. Thanks very much.